All right. Hello, everyone. Good afternoon, or morning, or evening, depending on where you are joining us from. Welcome to this session of Meeting Minds Global with Kellogg College. My name's Nicola. For those of you who haven't seen me before, although I recognise a couple of names today, I'm the Alumni Relations Officer at Kellogg, and I'm just going to quickly go over some housekeeping with you before we get started today. So if you can keep your microphones muted, please, for the duration of the talk, just so there's no noise interference, that would be much appreciated. There will be a Q&A session at the end where you can ask any questions that you have, which you can do out loud using the raise your hand feature or pop it in the chat box, where my colleague Sophie is keeping an eye out. I do also want to note that this session is being recorded, so whilst you are more than welcome to leave your video feed, to have your video feeds on if you would like, if you prefer not to, that's absolutely fine. Okay, so I'm going to introduce our speaker for this penultimate session in our Meeting Minds programme, and that is Professor Laurent Servais. Laurent is Professor of Paediatric Neuromuscular Disease here at the University of Oxford, and is an invited professor of child neurology at the University of Liège in Belgium. After graduating from Louvain Medical School in Brussels in 1999, Laurent completed a PhD in neuroscience from Free University of Brussels, followed by residencies in child neurology at the Free University and Robert Debré Hospital of Paris. In 2008, he took a position in neuromuscular disease and clinical research at the Institute of Neurology in Paris, where he was subsequently appointed head of clinical trials and database services. He joined the University of Oxford in September of 2019. Laurent has been the principal investigator in numerous clinical trials to test treatments for Duchenne muscular dystrophy and spinal muscular atrophy. He's also the leader of the newborn screening program for spinal muscular atrophy in Southern Belgium, and his main research expertise covers the development of innovative outcome measures, including connected devices for real-life patients' evaluations. Without further ado, I'll hand over to you, Laurent.
Thank you very much, and thank you for giving me the opportunity to present you some of the work I've been on over the last year, and some provocative thought. And the first provocative thought is certainly the title of this speech, which is not less than: to treat, to cure, or to make devastating rare diseases of children disappear. Is it actually just a matter of choice? Once upon a time, let's start with that. Once upon a time, there was a terrible disease. And that's the image of this terrible disease I've been educated with. That was we called flop infant, the Wernicke of men. These are kids who are normal at birth and who will very quickly in the first weeks of life present with weakness, and they will die for most of them. I've been educated with this image. I prefer that one. It's actually exactly the same disease, but look at the eyes of this baby. They are very special. The way they look at you, the way they interact with you. These are clever kids. And it's funny because it's actually one of the only disease we know in which potentially the patients are more intelligent than the average, and certainly the doctors. And this kid need a track, a permanent ventilation to be alive. These kids die before the age of 2 years for most of them, and some of them survive. And if they do, that's within terrible condition, wheelchair-bound, bed-bound, ventilated 24 hours a day. And I was lucky because we all have 1 or 2 patients in such conditions. But when we started the treatments I'm going to talk to you about, I had the opportunity to meet several of these patients, something like 20, 30 of these patients in this condition in a couple of days. And it was amazing to see that these are fascinating people who have fascinating life, doing fantastic things in their life. And I've learned from them that actually happiness and disability are 2 things that are completely uncorrelated. There is a milder form, if we can call milder a form in which you actually are able to sit but will never be able to walk. That's what we call spinal muscular atrophy type 2, because I forgot, by the way, to name the first form, which is spinal muscular atrophy type 1, the one that starts before the age of 6 months and in which patients die before the age of 2 years. So the milder form, if I can call milder form, in which you will never walk, is called SMA type 2, and it can lead to a normal survive in terms of years, but you are wheelchair bound and permanently ventilated at the end of your life. And you have a milder form, if you can call mild a form, in which you lose ambulation about the age of 10 years for most of the patients, which is called spinal muscular atrophy type 3. But the constant features across these different kinds is, look, well, look at this. Look at this way the patient is looking at you, and this easygoing attitude, this really smiling, seductive attitude. They will keep it across the different types and the different age. And which is quite incredible is that actually all these patients have the same mutation. It's all the same mutation in the same gene. It's a deletion of exon 7 of a gene that is called SMN1. And all these patients have bright eye, smiling, seductive attitude, but they have hypotonia, areflexia, few contracture, facial weakness only in 7 cases, and all have the same mutations, the same deletion in the gene, which is the exon 7 of gene SMN1. So why? How is it possible that patients who start late or patients who start early have the same deletion? It's because we have another gene which really looks like SMN1, it's called SMN2, but because of a very tiny mutation in SMN7 splice acceptor, this part of the gene is spliced, is removed nearly systematically. So there is very few functional protein that is read from SMN2. And because of that, if you have a lot of copies of this pseudogene SMN2, you may have a little bit of protein and then having a milder phenotype. That's why it's the same mutation that leads to actually a very broad phenotypic spectrum. So things have changed a lot. We got the first standard of care 2007, and then the second standard of care 10 years later with new treatments I'm going to talk to you about. And then came the question, which one and when? And of course, we came to the conclusion that we need early diagnosis, and for that we need disease awareness. And I'm going to talk about newborn screening, which is now leading to new standard of care. So first, the multidisciplinary management is extremely important. To treat this kind of patients, we need to be a team. You cannot do it by yourself. It's just impossible. It's very complex disease. You need to have a pneumologist on board, an orthopedist. You need to have palliative care, rehabilitation. You cannot treat these patients if you're alone. And that's thanks of this multidisciplinary care that our patients have grown. Some with type 1, the most severe form have reached adulthood. And I introduced you with my friend Marie-Antoinette in Paris. She wanted to have a baby, and she got it. And she got a baby. She is someone, very active on the social networks. So when a young girl with SMA type 1 or type 2 wants to have a baby or is dreaming about having a baby, she asks to Siri, hey Siri, is it possible to have a baby when you have a spinal muscular atrophy type 2? And then Siri says, you should go on the blog on Marie-Antoinette. So she goes on the blog on Marie-Antoinette, and Marie-Antoinette says, you should go to the consultation of Laurent Servais. And so at the end, I got several mothers with spinal muscular atrophy until I met this couple of patients with spinal muscular atrophy. And they told me a very interesting sentence, which was, with spinal muscular atrophy, everything is slower. Everything is more tiring. Everything is more complex. Going to school, to university, finding, keeping a job, a girlfriend. Everything is more complex, but everything is possible. And they demonstrated by having a baby. Of course, we gave a little bit of help on a genetic point of view, because as you might imagine, we didn't want this baby to be affected. So this baby is perfectly healthy. Look, they both lost 30 pounds in the process. And they take perfect care of this baby. That's the strategy that they had to sometimes to put in place, just for instance, to take this baby out of the park. and they are fully autonomous in taking care of the infant. So in this disease, we started 10 years ago, little bit more than 10 years, actually 15 years ago, the first trials, and it was the perfect storm. We start riluzole, it doesn't work. HDH, it doesn't work. Then TRH, it doesn't work. Gabapentin doesn't work. Creatine doesn't work. Valproic acid, doesn't work. We could add a little bit of levocarnitine, but unfortunately it doesn't work. Neither works sodium phenylbutyrate, salbutamol. Some say it works, but honestly, it doesn't work. And hydroxyurea, it doesn't work either. So behind all these drugs, all these trials, they are patients, they are investigators, passionate, they are clinical research assistant, but unfortunately, it doesn't work. And nevertheless, over the last 6 years, we got new generation of drugs working on either bringing a new gene into the nucleus of the cell, either interfering with the splicing process of this pseudogene SMN2, either working downstream on the muscle. And 5 years ago, was conducted a double-blind sham procedure control study, which means that we had a large group of SMA type 1 patients, which is the most severe form, the one that leads to death. And we stratified them in 2 groups. We were blinded. We didn't know which patient was receiving either the drug, either something that looks like the drug. And that was intrathecal injection, an antisense oligonucleotide, so a molecule that will interfere with the splicing of SMN2. And the basic question of this terrible trial was to figure out if we had more survivors in the treated group than in the untreated group, taking into account that when a patient starts SMA type 1, these patients will never acquire any milestone. They will just fall the cliff. So rather than showing you a lot of results, I can introduce you one of the kids of the trial. This young boy has obviously SMA type 1. Look the way he breathes, the way he moves. He has hypotonia, frog-like attitude, only small distal upper limb movement, no facial involvement, nice kid. In absence of treatment, these patients will die in a couple of months, and he will not progress at all. He will not make any motor progress. So we were blinded. I didn't know what drug, if this patient was on drug or not. And that's what happened 6 months later, that the same kid was able to turn around and to retrieve little toys and turn around again, which was something we never observed in patients before. Yeah, as you can see, the mother is very happy, and she's right to be very happy, because when you are able to turn around, that's extremely important during the night. Can you imagine if each time you have during the night, you have to turn around in your bed, you have to call someone to do it for you? This will translate into a major a burden for your quality of life and the one of your caregiver. And so that's the crude results of this study. We could find that the survival of the treated patients, the green one, was very significantly different of the patients who are not treated. And importantly, the patients who are doing the best were patients who are treated early, the patients with the shorter disease duration. So the patients who had the chance, if I might say, very soon after the onset of the disease to receive the medication. And the only factor that was associated with a better outcome was the disease duration. The earlier you treat this patient, the better was the prognostic in terms of survival, but also in terms of motor milestone. And this little gentleman was treated quite early. And this is the same boy a little bit later. He does something I was never be able to do, is to work without looking at the computer with your hands behind your back. It's useless, but look, he is doing that. And at the age of 3 years, he starts with the assistance, of course, of the mother to try to make some steps. Today, these patients have survived. They are doing better. They are progressing, but any of them is independent walkers. They remain with a significant disability. This boy, who should be dead today, is actually ventilated at night, is progressing, is a happy kid, by the way, and the family is happy, but nevertheless, he remains with a very significant disability. And the disability will be even worse if the patient has been treated late. If the patient has been treated after the age of 5 months, then the prognostic is not the same that if he has been treated early. But these patients, they don't walk. They survive, but they don't walk. And at the same time, and in the same edition of this major review, which is the New England Journal of Medicine, we had another approach, which was gene therapy, which consists in having the gene that is missing to place it in the vector and to inject a very high dose of the vector to get a transcription of the missing gene in all cells. And without entering into the details, we could see actually that we could get the same kind of results, which means that the patients who are treated get a much better survival, a much better outcome in terms of motor, but nevertheless remains with a significant disability. So that was transformative, that to be able to treat a disease and to be able to see kids who are normally dying, and I had seen these patients dying for years in my clinical practice, and not to see them dying anymore. That was a great progress. But that was not enough because these patients remain with a significant disability. And one day we start dreaming. And the dream came partially, at least, from this family. And this is patients of mine. That's the brother and the sister. And they are both affected, actually. The difference is that the mother was pregnant of the little girl, and she came with the brother to be treated. And she told us, we know that she's affected. Can you please treat her at birth, even before she develops the symptoms? And we did it. That's one of the first patients who are treated before the symptoms. And you can see the difference between this little girl now, age 11 months, who is completely normal, able to make some bodybuilding movement with the smartphone of the mother. And you can show this little girl to anybody, to any physician, any pediatrician, she's normal. And the brother is doing better. Now he's able to keep some kind of sitting position if he get the proper support of his hand. If I ask him to take my hand, look, he will do it, but it's so hard. And what happened later, because this was years ago, that was 1 year later. If you don't believe me that this little terror is normal, well, I don't know what to do. And this is the last video from this patients. The brother is still wheelchair bound and he is ventilated at night, but the little girl who has been treated before the symptoms is completely normal. And at the same time, I had another family. All the siblings was in the wheelchair, was treated. But this one was treated before the symptoms, was completely normal. So there was a proper trial conducted in 25 patients. And to make a long story short, patients with 3 copies of SMN2 have actually a normal development. And normal, I mean, it's not that they are better, they are normal. And patients with 2 copies, 50% of them are normal and 50% are mildly delayed. But when I say mildly delayed, it's not a matter of comparison with the other patients. So the gray here are patients who are not treated. The yellow are patients who are treated late. The blue and the red are patients who are treated just after the symptoms. And the blue and the green are patients who are treated before the symptoms. So no matter of comparisons, and don't believe that they reach a ceiling. It's just the scale, the maximum score on the scales on which we measure these kids with 26. So they cannot go above. But all patients with 3 copies are normal, and 50% of patients with 2 copies are normal. And with gene therapy is the same. Patients with 3 copies, they are all normal. That's the gray zone. The gray zone here is the normality, and all these patients fit into the normality. And for patients with 2 copies of this pseudogene SMN2, you can see that some of them are just below the normal. Most of these patients were identified because they were actually the brothers and the sisters of an affected sibling. So we knew as these genetic disorders that we had 1 chance out of 4 for the next to be affected. So then we start dreaming, and we start dreaming about newborn screening, which means to identify at birth kids who are affected and to treat them. And that's what we did in Southern Belgium with my partners in crimes, Tamara, Jean-Hubert, and François. We started a program that was called Sun May Rise, like SMA, and you can follow the story on Facebook. And March 5th, 2018, so 3 years ago, we screened the first baby. And actually, today, we have screened 130,000 newborns. So all newborns of Southern Belgium are screened, and we have identified and treated 9 cases. And these patients are doing perfectly well. So now, this project has been a success and has been fully implemented in the law in Southern Belgium. So now in Southern Belgium, all kids at birth are screened for spinal muscular atrophy. So which is fun in Belgium, by the way, is that we have not the same disease in the north and the south of Belgium, because newborn screening is actually a process that covers rare disease, but only with metabolic markers. And which makes this disease different is that it's the genetic disorders. The other also, by the way, but this one you can just identify by genetic. So it's a genetic test at birth. And the level of the threshold, as you can see, and the difference between affected patients in red or the others is very clear. So it's a PCR that we conduct on the blood of patients at birth on the Guthrie card. So this card that anyway is sampled at birth, and we can identify very reliably patients who are affected and to treat them. Then it's a genetic test. And as you might imagine, people can say, well, you know, this is eugenism. No, it's not eugenism. I mean, we're not selecting kids. We are just identifying at birth before symptoms patients we can treat and then to treat them. We don't identify carriers, which means that patients who have only 1 mutation, we don't know, and we don't identify them. And then, of course, you have the challenge of the predictive value of the number of SMN2 copy and the cost and budget impact. So from this, let me tell you another story, the story of John and Jack. And as you might imagine, that's just 2 names I invented. But the beauty of the story is that it's these 2 boys who were born approximately the same date. One was born in Northern Belgium, Jack, but he was lucky because the mother delivered in a Southern Belgium hospital. And the other one was born in France. And they both have spinal muscular atrophy. And they both were treated by gene therapy. And this is the little French patient. And he survived. He's a nice kid, lovely kid. He's not a walker. He has a good upper limb function. He's a painter. But look, he still has upper limb weakness. He's doing better and better, but he's not a walker. And he's a kid who remains with a significant disability. And this is John. This is Jack, the Belgian one. He has been identified immediately. We could treat him at day 41. And that on month 11, it's a normal kid. And a little bit later, this kid is actually a walker. Patients treated so far. And so that's the difference between one kid who is in the wheelchair, and you have to pay for the wheelchair. After having paid for the therapy, you have to pay for the wheelchair, you have to pay for the non-invasive ventilation, and the other who is normal. Actually, they've received the same drug, so it's the same cost. So the cost of newborn screening today is €3 per baby, which means that as it affects 1 kid out of 10,000, we need to spend 30,000 euros to catch a single kid who is sick. But then this kid will not need a wheelchair, will not need a non-invasive ventilation. So how much money do we save? Should we pay or not to pay? Well, let's make a calculation. You have 750,000 newborn in the UK. So it means that every year in the UK, we have 72 newborns with SMA. If we decide not to treat these patients, which is purely theoretical, because now we treat these patients, but historically, if we did not treat these patients, 44 will die, 23 will be wheelchair bound, and 5 will be walkers with difficulties. If we decide to treat these patients, but without newborn screening, we'll still have 19 will die, we'll have 38 in the wheelchair, and we'll have 15 walkers. But if we treat these patients and we have a newborn screening in place, we will still have the very early form that starts in utero and in which the patient is in such bad condition at birth that we cannot treat these patients, and 2 will die. And there are some forms that we cannot catch by newborn screening, very rare, and this will become sitters, but the other will be walkers and will be normal. Which means that if you compare these 3 populations, you can easily figure out that the most expensive one is at the upper right corner, with so many patients wheelchair bound. And the problem is that in absence of newborn screening, you're actually building a huge snowball of patients with disability. After 20 years, that's the number of patients you have in the wheelchair who will need non-invasive ventilation, standard of care, and it will be very burdensome for the social healthcare system. Which means that even if we don't care of curing patients, even if we don't care of having patients doing well, rather than having patients in the wheelchair, this is economically extremely important, taking into account that each of these patients cost about 7 million euros. So if you need 30,000 euros to avoid a patient that costs 7 million euros, then it's probably the best investment that a society can do. Of course, we are putting proper figures behind this assumption. I've got a PhD student working on this. I can tell you that the preliminary results confirm that screening saves a lot of money to the society. And that's a work that we just published in which now you can see that newborn screening is active in some countries, like one maternity in Russia, some province in Australia, 31 states in the United States, 2 regions of Germany, Italy, and of course, Southern Belgium. But then from that, we understood that we have actually several diseases that we could treat. We could treat much more efficiently if we treat early, but we don't screen this disease because they are genetic. And I will show you just one example, but I have many others. It's congenital myasthenic syndrome. This is a 9-year-old boy. He comes from Africa and is disabled since birth. He was wrongly identified as having Duchenne muscular dystrophy. Then a colleague of mine came and said, well, maybe it's spinal muscular atrophy. And then the parents contact me. And when he came to my clinic, if you know this disease, it's very easy to recognize a congenital myasthenic syndrome. And we could the same day confirm the treatment, the disease, and start the treatment. And that's the same patients exactly 2 days later. Just by giving salbutamol, which costs £7 per month. And it was 2 days. 2 days after the onset of treatment. And you can imagine that this boy needs rehabilitation. Nowadays is completely normal. The problem is that many patients have not the chance to get the right diagnosis. And we have similar examples in our centers with 2 patients, and one of them was made the newspapers titles. The other one was published in a journal, a medical journal. But we have other examples of the same mutations. And our 3 patients who diagnosed in Liège with this mutation, we have 100 cases in Oxford with really a center for this kind of disease, have spent together 70 years in the wheelchair, 20 years with invasive ventilation, 30 years with non-invasive ventilation, 5 muscle biopsies, more than 50 working years lost, and house adaptations. All of this could have been avoided. All of this could have been avoided by screening and by treatment of these patients. So you can imagine the amount of years that those human beings have spent with disability in the wheelchair, the money that is cost to the society, and all of this could have been avoided by newborn screening. And we don't screen this disease just because it's genetic. So my vision is that actually genetic newborn screening, which is exactly the same concept of non-genetic newborn screening, but using the technology of today, has the potential first to improve current screening. And I will not go into the details, but for instance, central hypothyroidism is not screened today by the current newborn screening. We could do it with genetic. We could screen the disease that we can treat today, and we will be able to screen the disease we will treat tomorrow. And there is no treatment, there is no screening today for all this disease, which means that we are developing drugs that will at best let patients live life, but which will never cure these patients because their brain or their muscle are destroyed and we arrive too late. This is the calculation I did for the UK. For the main disease we could screen if we go to genetic newborn screening, and the number of patients per year in the UK, 241. 241 patients per year in the UK could be screened and treated at birth today for diseases that are perfectly treatable. Genetic central hypothyroidism can be treated with thyroid hormones, which is extremely cheap. Dopa-sensitive dystonia is treated by dopamine, which is very cheap. Congenital myasthenia is treated by pyridostigmine or salbutamol. It is extremely cheap also. Brown-Violet of Venlar is treated by thiamine, which is incredibly cheap also, which means that for many of these diseases, actually, it's even not a matter of price of the drug. And we could also screen in other countries where the healthcare is not so developed. So a dynamic genetic newborn screening could help to accelerate the development in the rare disease field, to improve the cost effectiveness of treatment, and to save lives and decrease disability. And for the UK, we talk about 200 kids per year. There are obstacles. The first is that we are conservative by nature, and we consider newborn screening as a metabolic test, and we need to change our mind and to understand that the times have changed, and there are several treatable conditions that we cannot identify by a metabolic test. There is the fear of eugenism. When you speak about genetic newborn screening, this is not eugenism. These are kids who are already born, and the question is to wait them to be symptomatic and to wait too long and to treat too late, or to treat them when they still have a chance to be normal. And of course, as usual, initial fundings, because this needs equipment, this needs people and midwives to consent the mother. The good news is that in Belgium, I started a small program on just one maternity, but this kind of program, as you might imagine, is extremely expensive and require fundings that you cannot just get by a single grant. So that's where we are today. We are living exciting time because we are now able to treat diseases that we were not able to treat before. But we should not just treat the disease. We should try to make it disappear. And we can do it. Spinal muscular atrophy is the best example. There are many others. It just depends on the strategy that we put in place to try to get the best from this innovative medication, and not just to keep our patients alive, but to give them a normal life. So if I want to take 1 or 2 steps back from this crazy period of 2007 to 2021, and try to figure out this period into a much longer period of time with 2 very important dates, 180-90 and 2045. The first is the description of Spinal Muscular Atrophy by Guido Wernick and Johan Hoffman. And the second is my retirement. And I hope to be, kind of old man telling to my granddaughter, oh, when I was a young child neurologist, I still have seen kids dying of SMA type 1. And I really hope that answer will be, could you please give me a break with your old stories from the past? This does not exist anymore. Actually, we could fill cemetery with disease rather than with kids, and polio has disappeared. Today, we have the possibility to make virtually SMA disappear, but we have learned a lot, and there are several other diseases that could disappear if we are clever enough to have the right process and to accelerate the development by genetic newborn screening. So that's just the end of the talk. And as you might imagine, all of this work can be done only with teams. My teams in Paris that I've left, the team in Belgium where I still go, and my new team in Oxford with my partners in crimes for newborn screening in Southern Belgium. Thank you very much for your attention, and I would be happy to take any of your questions.
Laurent, thank you. That was just fascinating to hear about your work, and that there really are answers to some of these things that cause such distress to children and their families. So thank you.
My pleasure.
I can see a hand up already. Sue, hello. Sue, you have your digital hand up. Would you like to unmute yourself so that you can ask Laurent a question? Oh, hand is down.
Sorry, it was meant to be a clap rather than a hand.
Oh, I'm sorry. Oh, Sue. Thank you.
Really wonderful, inspiring talk. Thank you.
Well, actually, I have a question, if I may, Laurent. Thank you very much. Obviously, I'm not a medic. I really knew nothing about this until listening to your talk just now. But your point about the obstacles, I'm just wondering how can you medics bring about a change so that this screening strategy could be put in place? I mean, is it a change in government policy? Does it require lobbying? I have no idea what the process between you and the medics and a policy change might be.
Well, it's a little bit of everything. Just to share with you my experience in the UK. When I came in the UK, I said one of the first things I wanted is a newborn screening for SMA, because when you've been living 3 years with newborn screening with SMA and you don't see this horrible disease anymore, you said, no, I want to go back 3 years back and I want to implement newborn screening here. This is incredibly difficult. So I could get the funding for the screening for newborns for SMA in the Thames Valley. We wrote a protocol, and then what we just wanted is the authorization from the newborn screening committee to use part of the dry blood test that is actually thrown away if we don't use it. And they just told us, no, we need to discuss, and you need to discuss with that subcommittee, and you need to. So we are lifting, one by one the obstacles, but we face a real bureaucracy, that's for sure. And we need to be honest and transparent on that. This is not easy to put in place. That's the first thing. The second one is that there is, in other countries, because we are a very international audience, like in France, for instance, they made a kind of big basket, what they call the bioethic law, when they discussed all together carrier screening, newborn screening, mother who are pregnant for another couples, and so on and so on. So it was, a complete nonsense. And the people who were elected, the deputies, they don't have the knowledge. And so they were just confused and influenced by lobbying, religious lobbying. And at the end of the day, it was the perfect storm. So I think that the very important point first is to educate and to explain that this is newborn screening. Those kids are born. It's not to select people with blue eyes. It's to detect diseases and to treat this disease, nothing else. I mean, we don't want to create a new race without disease. No, we just want to treat the disease. That's it. And then the next point is obviously funding and the cost. The initial pilot that we are just starting in Belgium for very small maternity is 2 million. If we wanted to have a similar program as this with a similar size in the UK, that's more something like 10 or 15 millions, which altogether is nothing, because it's when you compare with the benefit, it's nothing. But when you have to put this money all together from different sources, because there is no agency that funds 10 million for a single site, that becomes a very different stories. The good thing is that, and that's why I'm a little bit more confident in Belgium, is that we can actually start very broad newborn screening for 143 diseases. So that's a lot of diseases. And my hope is that I can attract from the pharmaceutical fundings, just because we have a project that has started. But of course, for me, that's the next step in the UK. And I need now to start my SMA newborn screening to show that I'm not disruptive, that the newborn screening committee in the UK is not collapsing because of my project. And when you have demonstrated that you're not disruptive, to gather the funding and to expand it to all these other diseases on which we should be much more efficient.
Thank you. I can see we've now got some questions coming up in the chat. May I? We've got Sue asking, what is your view on prenatal screening of embryos for parents having IVF?
So I think that there are several different kinds of screening, and it's very important to make the difference. There are parents who are, for instance, who have already a kid with spinal muscular atrophy, and they don't want to live another time such a devastating disease, which is perfectly understandable when you know what means this disease for parents. So it means that then you have the possibility either to have in vitro fertilization and to select an embryo that is not affected, or to initiate a normal pregnancy and then to test if the fetus is affected or not, and then to make the decision according to your philosophical opinions. For me, both options are valid, and personally, I can only empathize with parents who have lived the terrible adventure of being the parent of a patient with spinal muscular atrophy. So, so that's the pre-implantation screening. But I'm just speaking about a disease, right? I'm not speaking about the color of the skin, the color of the eyes, the strength, the age. I'm just speaking about a single disease. And the next point is, and this is also something that has really started in the US, for instance, but also in Belgium, is that you can also propose to anybody who wants to have kids to see if you are a carrier of this terrible disease. And if you are a carrier and you, the person with whom you want to have kids is also carrier of the same disease, then to reimburse in vitro fertilization and to avoid the kids to be affected. That's what we call carrier screening. This is a complete different process. And in Belgium, we have both. We have carrier screening and newborn screening. But there are a lot of differences. The first one is that carrier screening is really socially oriented. As you might imagine, there are young mothers who have never discussed with a doctor before being pregnant. And sometimes the father is not there anymore. So they have not been to a doctor's, and pregnancy was not a project that they did together. So it's very socially oriented, first point. Second point is that we know that 5% of kids are actually not the biological son or daughter of the father. So which means that carrier screening doesn't work there. Then you have also what we call neo-mutations, so which means mutations that happen in the kid, but the parents are not carriers. And there are also other cases. For instance, when the parents have two normal copies of the gene on one chromosome, but no normal copy on the other chromosome, that you cannot identify by carrier screening, which means that the beauty of newborn screening is that you just take all kids, regardless if the parents are rich, are poor, are socially educated or not. It's really egalitarian. So I personally think that giving people the possibility, if they wish, to avoid having a baby with a severe disability is fair if they are properly informed, and if they agree, if they are happy to go for in vitro fertilization afterwards, why not? But we must be conscious that it will not make disease disappear. It will just decrease the number of cases and decrease the burden and the cost, which is fine, but it's not enough. And we need to give all kids the same exact chance. So that's why I think that carrier screening is a possible strategy, and it causes other problems, because you can imagine that if you tell me, well, you're a carrier of that disease, it's not a problem. The problem is that your wife also, then you can look at your wife and say, are you the right person to have children with? Of course she is, if you're in love with her and if it's your project, but you start inferring with the personal life of people by telling them that to a certain extent they are not compatible. So this is a completely different story, and carrier screening can be an option if you are very well informed, very well educated, if you are able to understand all the consequences, but it will not cover the entire population like newborn screening. So both may happen together. So that's the different level of screening. Carrier screening before even the pregnancy is initiated. In vitro fertilization with selection when a patient has been already identified and you want to avoid it, or antenatal screening when you are already pregnant and you want to know if the fetus is affected, and then newborn screening. And I think that all options are valid and could work together to decrease the number of cases, and when the cases happen, to be treated as soon as possible.
Thank you. And Rob is asking, does the treated child pass on the SMA to its own children?
Actually, if they married with someone who is affected, the answer is yes. So the couple that I've presented to you is a special story, because of course we assist them and they could get an embryo from another lady. So at the end of the day, the children was not affected. But if, let's imagine, I've got an SMA and I get married with someone who was not an SMA and she's not a carrier, there is no chance to give the SMA to my children. So the answer is only if both parents are affected, which might happen, but then we know it and we give a little bit of help to avoid it. But I have a lot of mothers and ladies who have children, but the father is actually healthy, so there is no problem for the kids.
Thank you very much. We've got time for probably one more question, if anyone has another question for Laurent. I'm just checking to see if I can see any digital hands coming up. No? No? Stunned into silence, Laurent. Thank you very much.
Thank you. It was a pleasure, really. And I really hope that we will meet all together in Kellogg College in some months.
Yes, so do we. And I can see Nicola there just come back onto screen to close the session for us.
Yes, yeah, I'm happy to wrap it up here if nobody's got any last minute anything that they want to say. Okay. Well, in that case, thank you so much, Laurent, for your talk this afternoon. That was that was really inspirational. I wasn't sure what to expect from this with my sort of lack of medical knowledge, but I think it's amazing the work that you've done and the work that you're doing, making such a difference to these children. And thank you for sharing that with us. Yeah, and thank you to everyone who's come to listen this afternoon. I hope you've been as inspired as I've been. And yeah, if nobody's got anything else, then I will end the session here. Thank you all very much for coming. Have a good rest of your day, wherever you are. And yeah, I will hopefully see you all again our next session, or in the future, maybe even in person.
Who knows? Who knows?
Bye-bye.
Bye, everyone.
Thank you.
Bye-bye.
Thank you. Bye-bye.
Bye-bye.